So we're ready to go. Hello, everybody. Good evening. Welcome to the University of Warwick and our Science on the Hill online public event on COVID-19, on everything you've ever wanted to know, but daren't ask about COVID-19. My name is Lawrence Young, and I'm a professor at the medical school at the University of Warwick, and I have a background in virus research. Science on the Hill is supported by the Warwick Medical School, by our School of Life Sciences at the University of Warwick, and by our quantitative biomedicine program. As I hope you're all aware, we've previously held a series of these public science evenings as in-person events on campus, but unfortunately, due to current circumstances, obviously we can't do that. But we thought this was a great opportunity to reach out and share our research, our expertise, while you watch from wherever you are. So the evening is going to start with me giving a brief overview of COVID-19 and the virus that causes the disease. And I'll talk a little bit about vaccines. Then Dr. Nicole Robb will discuss her latest work on detecting the virus and the development of a quick test. And we all know how important mass testing is to help us get out of the current problem we have with this virus. And then Dr. Chrystala Constantinidou will discuss how we can track the spread of the virus by studying the changes in the virus's genetic code. And that's a really important way of mapping where the virus has come from and where the virus is going. So, as usual, there'll be time for questions and answers at the end of the of our presentation. So please use the chat box function as we go along and put any questions there. Any comments, any questions, tell us where you're viewing from, and then we will be able to select some of those at the end and have a nice session where we will try our best to answer questions. As people are aware, this is a very fast moving field, but I think we're pretty up to date with what's going on, and between us I think we can probably cover most of the questions, but we really look forward to hearing from you all. So thank you for joining us, and I'm going to start now by sharing my screen. Okay, so let me try and go backwards. Right, so okay, so I just wanted to start with where we're at today, and this is a screenshot from the World Health Organization, and they update the statistics on infections every few hours, actually. And so the frightening figure that we reached today was over 50 million confirmed cases. Actually, we turned 50 million a couple of days ago. 50 million confirmed cases of COVID-19 across the world, including, as you can see, a large number of deaths. And so we're really still struggling with this virus, and we still need to think about how we can manage it, even with the hope of a vaccine. I wanted to talk a little bit about what viruses are, because they're rather strange things, and there's always been a bit of a debate about whether viruses are actually alive. What type of life are they? They're really a bit like zombies, because in themselves they cannot grow, they cannot develop, they cannot replicate and grow themselves. The thing a virus wants to do is just to grow itself and pass itself on to other cells so it can keep growing. So they're a bit like zombies. They need to live on us and other animals to grow. Okay, so this is a slide that talks a little bit about the nature of viruses. So they're not like bacteria, they're much smaller. And as you can see from that picture, that cartoon, at the bottom there's a very large drawing of what a bacteria looks like. And a bacteria actually can do everything itself. It can grow itself, it can feed itself. Viruses can't do that. And what you see above that picture is a load of different types of viruses, cartoons of the different shapes and sizes of what are an amazingly large family of different viruses. This thing's got a life of its own at the moment. So anyway, what you can see here is different types of virus, and so these are really on the edge of life. They're not living. They feed off us, and their main reason for existing is just to grow themselves, to replicate themselves, and then for that infection to be passed on to other cells and other people. So they're very strange things, viruses. They all have a very similar structure. So this is a cartoon version of what a virus actually looks like. And it's particularly relevant because this is what the SARS-CoV-2 virus, the virus that causes COVID-19, looks like. It's got an outer membrane, which is a fatty membrane. And then inside it's got a crispy protein coat. And inside that protein coat, it's got nucleic acid. This is the genome of the virus, and the genome of the COVID-2 virus is an RNA genome. That doesn't really matter. What matters is what you've got here is a protective coat called the capsid around the genome, and then the whole thing is enveloped in a lipid membrane. It's actually that lipid membrane that makes the coronaviruses very susceptible to soap and water, and indeed to alcohol, and why washing hands is so important. And this is what the virus looks like under a microscope, under an electron microscope. And the important thing that we're going to come back to is this crown of these proteins on the surface of the membrane. This is called the spike protein. It's really important, and it gives it this crown look, and that's why it was called originally a coronavirus. When the first coronaviruses were identified and looked at under a microscope, electron microscope, at very, very high magnification, you can see these spikes. And on the right-hand side, you can see what this looks like in a cartoon form. Those red blobs are the spikes that stick out of the virus, and they give it this sort of crown effect. So this is a complicated slide, but I thought it's worth showing for a couple of reasons. At the bottom, again, you can see a cartoon version of what the SARS-CoV-2 coronavirus looks like. It's got an RNA genome that's encased in a protein, and it's got these spike proteins. And this is the genome of the COVID-2 virus. This is what encodes all the different proteins and things the virus does, mainly to replicate ourselves. And this is a 30,000 base pair long piece of RNA. Essentially, it encodes all the instructions to make the virus, and it's about a millionth the size of our own genome. And we're going to hear from Chrystala a bit more about some of the variations in that genome and how that might influence the way the virus behaves. I'm not going to go into too much detail about that just now, other than to say this is the way the virus parasitizes us. It feeds on us, it infects us, and its main reason to doing that is to replicate. So here's a diagram, which in fact, we use this to teach the medical students, because this is the basic thing that viruses do. Viruses bind to the surface of one of our cells. For the SARS-CoV-2 virus, these are cells in our noses and throats. The virus binds in a sort of it's like a lock and a key, if you like. The virus locks onto this key on the surface, and then it gets into the cell, and then it takes over the cell that it infects and converts that cell into a factory to make more viruses. So it's quite frightening, because it basically kills off the cell in the process of growing itself. And that's what happens when you get a cold or when you get flu. It's exactly what happens with other virus infections. They all have this similar approach to getting into cells and then to growing and replicating themselves. So here's somebody who is infected with the SARS-CoV-2 virus, breathing it in and breathing it out. Of course, when you breathe it in, when you get infected, and we know that infection is through droplets, through nasal droplets and through aerosols, it infects our nasal passages, grows in those passages, as I've just shown you, and then it percolates down into our lungs. And this is where the virus causes problems, because it gets into the lungs, and then it causes inflammation in the lungs. It causes secondary infection, which results in pneumonia. And that's what COVID-19 is all about, really, that terrible pneumonia. Now, the reason I focused on this spike protein is two reasons, really. This spike protein on the surface is the lock that fits into the key. It's the lock that binds to a protein on the surface of cells, and it's that allows the virus to get into cells. So understanding this spike protein is really important, because we know it binds to this receptor, and if we can block this lock and key mechanism, we could block infection. And there's a lot of work going on to develop drugs that do that. And here's a blown up picture of what that looks like. And the other reason for being interested in the spike protein, without going into too much detail, is that the spike protein, we know, can stimulate the body's immune system. And when the body's immune system kicks in, antibodies are made which can bind to the surface of these spikes and stop the virus infecting. And also there are other types of immune response that can also protect against infection. So we know that an infected individual can develop these immune responses, and we are excited about the possibility that vaccines will do the same thing. When somebody's infected, it takes about 5 days for them to get symptoms, and once they develop symptoms, then the virus continues to grow. This is a graph of that, and as the virus is growing, the body's immune system kicks in. This is the antibody response, and it's usually around 10-11 days that people who are going to get sick get really sick, and they get so sick sometimes, unfortunately, that they need to go onto a respirator to give them oxygen. Okay, so in the middle section here is just a little bit about how infection works. We know that this virus originally came from bats. It probably went through another host. This is a pangolin, and then it somehow got into humans. And we know that we can spread this through small droplets and large droplets. We know how important direct contact is. That's why hand washing is so important. And we know the virus can also stay and live and survive on different surfaces. So that's really important in terms of being careful about what you touch, when you touch, and where you go. So we're learning a lot about this, and it's why it's so important to wash hands, to wear a mask, and to keep distant from people. Okay, we'll hear a little bit more about that later. Now, the thing that we're just learning about this virus is not only does it infect the lungs and cause all sorts of problems associated with the typical sort of symptoms of COVID infection, but it can also get into other tissues, and it can cause problems with your kidneys. It can cause problems with liver, and we are learning more and more about how the virus can also cause problems with your brain. Actually, it can infect brain tissue, it seems, which is quite surprising, and we're learning more about that. And that's what you hear about recently when we talk about long COVID, that people who recover from the acute infection still suffer a number of other symptoms. And we know that all of this is made worse if you have other diseases. So if you have cardiovascular disease, if you have, that is, high blood pressure, or if you have diabetes, everything is exacerbated by this very, very nasty virus. I'll just rush on now. So basically, you're going to hear a bit shortly from Nicole about detection. Detection diagnosis is really important. We're at long last getting to grips with testing in this country and tracing and isolating. Obviously, there are loads of different drugs that have been going through different testing at the moment. Some of them, dexamethasone, for instance, looks really useful in patients who are sick in hospital. And then obviously we've heard, particularly in the last 24 hours, about vaccines and the big rush to develop an effective vaccine. And so here I've just got these three areas. There's a whole lot of work going on, taking old drugs and trying to use them or repurpose them for use in this disease. And we're going to hear more about that in the next few weeks as some of the clinical trials report. There's the possibility of using antibodies to treat patients, either antibodies from the blood of folks who've had infection and recovered, that's called convalescent plasma, or modified, artificially made antibodies, which apparently were used to treat President Trump when he was sick. And then, of course, there's vaccines. And what vaccines try to do is to trick the body's immune system to think it's being infected by the virus, but it's not. It's being exposed to bits of the virus. So here you can see a number of different approaches to vaccines. We're used to vaccines being a weakened form of the whole virus. If you think about polio vaccine or measles vaccine, they're weakened forms of the whole virus that you get infected with that doesn't give you the disease, but fools your body's immune system into thinking you've had the disease. What we heard about yesterday was a really unique and novel approach where you don't take any of the proteins or the whole virus. You take a bit of its nucleic acid that makes the spike protein, and you trick the body's immune system into generating an immune response to the spike protein. And that's a really exciting development. So one of the things that clearly is happening at pace, at warp speed, is the development of clinical trials. We know that in the classical development of any vaccine, making sure it's safe and is properly tested in large numbers of people and it's manufactured appropriately can take up to 10, 15 years. What we're seeing at the moment is this fast tracking of the COVID-19 vaccine development. And what's really important is while you're doing that, you must make absolutely sure that the vaccine is safe. At the moment, there are different phases of this whole clinical trials pipeline, and there are about 35 vaccines being tested in early trials and 11 of them in later trials. 6 have already been approved. That's a bit controversial because they've been approved in countries like Russia and China, where we're not convinced they are safe enough. Okay, so my final bit here is just to say, well, this is the vaccine we heard about yesterday. It's an RNA vaccine wrapped up in a fatty coat that again tricks the immune system into working, and you can inject that and get an immune response. And that immune response is predominantly to this spike protein on the surface, and that spike protein is really important, as you've seen earlier, in the way the virus infects our cells. So if these antibodies interact with the surface of the virus, they can block infection. And that's what we saw in those preliminary results that were announced yesterday from Pfizer, that this new RNA vaccine can generate a protective immune response that seems to be extremely useful in preventing the COVID-19 disease. And perhaps we'll talk about that a bit later. I'll now hand over to Dr. Nicole Robb, who's going to talk a little bit about some of her work in developing rapid tests that are so important for the diagnosis and the tracing of infected individuals. So thank you for your attention.
Right, good evening, everybody. So I'm Dr. Nicole Robb, and I'm going to be talking a bit about how we test for the coronavirus. So widespread and accurate testing is really, really important. We need it to help us keep track of the number of cases, and that helps us to guide our response to the pandemic. We also need to quickly identify cases so that those people that need it can be treated, and that people who are ill can isolate. And this all works together to help us prevent the spread of the disease. There are 2 main types of coronavirus test. So antigen tests can tell you if you currently have an infection, and antibody tests can tell you if you've had an infection in the past. So to understand these tests in a bit more detail, we need to look at the SARS-CoV-2 virus itself. So this is a small circular virus. It's got a fatty lipid membrane wrapped around it, and protruding out of that lipid membrane are proteins that are called spikes. In the middle of the virus is the viral genome, which is made up of a nucleic acid called RNA, and this is wrapped up in another viral protein that's called the nucleocapsid. So all of these structural parts of the virus together are called antigens, and antigen-based tests are designed to detect these structural parts of the virus. So I've mentioned the spike protein, and Lawrence also described earlier how important this part of the virus is. So this is the part that binds to the receptors on the surface of host cells and allows the virus to enter your body. But one of the ways that our bodies fight the virus is by making small proteins called antibodies. So these work in lots of different ways, but one of the ways that they can help prevent infection is by binding to the spike protein and blocking the virus from entering host cells. It's these antibodies that are detected in antibody-based tests and can tell you if you've been exposed to the virus before. So there are different types of tests, and they're all different they're all useful at different stages of the disease. So in this graph, we've got the time from when you're first exposed to the virus in weeks, going from left to right. So the first type of tests that I'll be talking about are these rapid antigen tests. And these are really useful in picking up when people are actively sick with the virus. So in the first week or two after they've been exposed to the virus, when they've got virus in their body, these tests are useful for picking up parts of the virus and giving a positive result then. There's also PCR-based tests, which are also antigen-based tests, and these work at the same time as the rapid tests, but they can also detect small fragments of the viral genome long after people are not infectious. So that's why there's this big long tail off going all the way up to around 4 weeks after you first get symptoms. And finally, there are antibody tests. So for SARS-CoV-2, you don't get antibodies until at least 1 to 3 weeks after you've first been exposed to the virus. So these tests are not useful in a diagnostic sense, but they are really, really useful in giving us an overall picture of how many people in a given population have been exposed to the virus. So I'll describe these 3 types of tests in a little bit more detail. And we'll start with PCR, which stands for polymerase chain reaction. So this is an antigen test. It works by amplifying up the viral RNA. It's often called the gold standard for COVID testing because it's very, very reliable. So it requires a specialist operator to carry out the test and specialist equipment, and it needs to be done in a high containment lab. So this is great, but what it means is that the tests can be pricey, and they can also take quite a long time to give you the results back. So you can wait around 2 days to hear if you've got coronavirus with a PCR-based test. And like I mentioned before, it can also detect RNA in your body after the virus, after you're not infectious anymore, so it can give false positives. So how this test works is you have a nose or a throat swab, and RNA is extracted from the virus particles that are present on the swab, and that RNA is then copied into its more stable cousin, which is DNA. The DNA is then heated and cooled many times, and that allows enzymes to copy the DNA and make lots and lots of copies of it. As each new DNA copy is made, it incorporates a fluorescent dye. So as the DNA gets amplified, you also get an increasing amount of fluorescence in the sample. And this is used as the readout for the test. So if you have a test with no virus in it, there's no DNA there to be amplified up, so you don't get any increase in fluorescence. If there is virus, though, there in the beginning, that genome can then be amplified up, and as it does so, you get an increase in fluorescence, and once this passes a certain threshold, the test is positive. So that's how PCR works. The other type of test that you've probably heard a lot about in the news in the last couple of days are rapid antigen tests that are based on lateral flow. So these are also antigen tests. They tend to use a liquid sample, so something like saliva. They're very, very quick, as they can be done in around 15 to 20 minutes. They're relatively cheap, and they don't need a lab. So this sounds absolutely perfect for mass testing, but there is a small caveat because they're not always accurate. So they can be very accurate, so about 90% accuracy in cases where people have a lot of virus in their body and just after they've been exposed to the virus. But if you have cases where the people have much less virus, so for example, they're asymptomatic, or you're testing, for example, a week after the person's been exposed, then the accuracy of these tests goes down to as little as 60%. So they tend to work on something called a lateral flow format. And this is a lot like a pregnancy test. So it's normally a little cellulose-based stick. The sample, which can be saliva, which might have some virus in it, is inserted at one end of the device. And then there are antibodies inside it that basically are designed to bind to parts of the virus, so the viral proteins. So the first type of antibodies that are in there are designed to recognize viral proteins, but they're also labeled often with something like a gold nanoparticle, which means that they're visible to the eye. So what happens, there are other antibodies further down that I'll talk about in a second. So what happens is the sample gets put in, and it then flows from one side of the device to the other, so from left to right on the screen. And if there is virus present, it's bound to the first set of antibodies that also contain the gold nanoparticle. They then continue to flow through to the test line. So these antibodies also bind to the virus if it is present. And because of the gold nanoparticle on it, you then get a signal on your test line. And then finally, there's something called a control line, which basically tells you that the sample you put through at one end has flowed all the way through past the test antibodies. And these are antibodies that, instead of detecting the virus itself, they bind to part of the initial labeling antibody. So you will also get a signal in the control line if the test has worked properly. And in this example, it's a positive test because there was virus present in the sample that was put in. The last type of test are antibody tests. So these are blood tests that look for antibodies against the coronavirus. Like I said before, it takes between 1 and 3 weeks for these antibodies to reach detectable levels. So this is useful to tell you if you've had an infection in the past. There are different formats for these. So these can be done in batches in a laboratory, or they can have a lateral flow format like the one that I've just described, and it can be used in community testing or at point of care. So our approach in the lab is a little bit different. We have been working on viral detection and viral diagnostics for about the last 4 years. But since about April or May time, we've basically been working flat out to try and take what we know and develop it into a COVID-19 test. And so what we've come up with is a 5-minute antigen-based test. But rather than working to detect small parts of the viral protein, of the virus, like viral proteins or viral RNA, we actually detect intact virus particles. How we do this is we have throat swabs from patients, and we label the virus particles in the sample, and then we use a microscope to look and see what's in there. We then get lots of microscope images, and we have an artificial intelligence-based software that looks at those images and can tell us if a virus is present there or not, or even what type of virus there is. So this is really neat because what you can do is you can train the artificial intelligence. So you could show it pictures of what a sample that has a virus in it looks like, and then you can show it pictures of a sample that doesn't have a virus in, and it learns what those looks like. And then when you show it a completely new sample, it can tell you what the result is. So because we're looking at intact virus particles, that has a number of advantages. So we don't need to extract the viral RNA. We don't need to amplify up anything. We don't need to purify the sample. We just work directly on the sample as it comes out of the patient. So all of that means that the test is really, really fast, and we don't need any specialist kind of lab or specialist operators to carry it out. So in a bit more detail, what happens when we get the throat swab from the patient that has virus in it? We need to make that virus fluorescent so that we can see it with a microscope. So to do that, we use a little trick that we discovered a couple of years ago that takes advantage of the fact that the virus is covered with a lipid membrane, and that lipid membrane has a negative charge. We then take short DNAs that are fluorescent, and these DNAs by their nature also have a negative charge. We then take a positively charged solution, and that acts to basically bind the negatively charged DNAs to the negatively charged virus particle. And this is really neat. It works really, really fast, and it results in small small virus particles that are very, very bright and can be easily seen on a fluorescent microscope. So we do this labeling. We use a microscope to take lots of images of a sample very, very quickly. So we end up with hundreds of different images. We then use computer software to pick out the individual signals in each image. So each signal here corresponds to a single virus particle. And so we end up with thousands and thousands of these little tiny pictures, each one of a virus particle. And we feed that into our machine learning neural network, and that's able to tell what's what. So we get a result back within 5 minutes. It can tell us if the sample is positive or negative for coronavirus. And it can even go a step further than that. In a positive test, we're able to tell the difference between SARS-CoV-2, which is the virus that causes COVID-19, and other seasonal coronaviruses that are going around, as well as influenza. So this is really important at the moment. We're going into winter. There's a lot of other respiratory viruses co-circulating around, and it might not be possible initially to tell why somebody's sick. So our test is really unique because it can quickly and easily tell the difference between these different viruses. So to summarize, testing is really, really crucial. We need it to help prevent the virus from spreading. There are lots of different types of tests, and they all have different uses at different points of infection. So we've developed an exciting alternative where we use imaging and AI for viral detection. And using this test, we can test very accurately within just a couple of minutes. So this is a massive group effort. I have to say thank you to all the members of my group, and especially Nicholas Shilas, who really led the work. We've had lots and lots of help from clinicians, from people lending us microscopes, from academic collaborators, and from all of our funding sources as well. And thank you for listening.
Okay, thank you. Thank you very much, Nicole. And please stay with us, and please put any questions you have in the comment box. It's a great opportunity for you to ask us difficult questions, or perhaps not so difficult questions, but please do put any questions in the comments box. And we're going to hand over now to Chrystala, who's going to tell us a little bit more about how you can use sequencing of the genome of this virus to trace the spread of infection. So over to you, Chrystala.
Hello, thank you, Lawrence. Let me start my presentation. Okay. Hello, so I'm Chrystala Constantinidou, and I will be talking to you about how we can use sequencing to understand transmission patterns of organisms such as viruses and bacteria. And of course, I'm going to talk to you about the SARS-CoV-2 virus and its sequencing. And I'm going to talk to you about a small set of samples associated with the care home that we sequenced here in Coventry. I will start with some basic concepts you probably all know already. DNA, as you've already heard, is the hereditary material that is present in humans and almost all other organisms. It is made up of four subunits: adenine, cytosine, guanine, and thymine, and these are also known as nucleotides. They form two ribbons that fit together like two rungs of a twisted ladder, and this is the structure that we all know as the double helix. Specific segments of the DNA are called genes, and these serve as templates to make RNA. Now, the information contained in the RNA is then translated into proteins, and these serve as the building blocks of the organism. The collection of all genes in the DNA is called the genome. Now, when a cell divides into two, it needs to replicate its DNA so that both the original and the daughter cell end up with a complete copy of the genome. Now, this is not an error-free process. During replication, often nucleotides are misincorporated introducing what we call mutations in the genome. When this happens, we end up with one cell containing the original genome and a new cell that contains a genome with a mutation. Now, we can follow these mutations, these changes as they emerge, and this enables us to determine how closely related the two cells are. The mutations happen in random positions. Not all positions affect the nature of the protein that is produced by that specific gene. The rate of mutation varies amongst organisms. This is dependent on things such as the division rate of the organism and on the fidelity of the proofreading enzymes that are associated with the enzyme we're studying. Sequencing is a process that allows us to determine the order of the nucleotides in the genome. The first sequencing protocols were first established in the mid-70s, and they were very slow, cumbersome, and they used lots of nasty chemicals. And although during the next 25 years improvements were introduced, it still took us 10 years, hundreds of scientists across the world, and millions of dollars to sequence the genome of one human individual at the turn of the century. But disruptive advances were made in the way we do sequencing, and in less than 2 decades, we can now sequence multiple human genomes in a single lab in a couple of days for just about $100 each. So the potential of such data being at our fingerprints has made science very exciting and led to new areas of biology, such as genomics. So how can we use genome sequences? Here's an example of, let's say, 5 genomes of a bacterial species isolated from 5 different patients in a hospital. Now, the circles represent mutations, and each color represents a different mutation. So all 5 have the same blue mutation. 3, A, B, and C, contain the yellow mutation, and 2 of these also contain the green mutation. A and B also contain the green mutation, sorry. A and B have identical genomes. D and E contain an additional red mutation, and C and D also contain unique mutations, the gray one for C and the orange one for E. Now, we use special software and algorithms to look at the millions of nucleotides in each genome, and we construct what we call phylogenetic trees. These show us graphically which genomes are more closely similar. So genomes on the same branch of the tree are more closely similar to ones on different branches. So in this instance, A, B, and C are all on the top branch, and D and E are at the bottom. Also in the top branch, A and B, which are identical, form their own sub-branch here. The distance of the branches from left to right can represent either time between mutations or the number of mutations. So from this information, we can build transmission trees. It is clear in this case that E has originated from D, and in that process, it has acquired the orange mutation. It is not clear whether A originated from B or the other way around, but we can deduce that all 3, A, B, and C, originated from an unknown source that we haven't sequenced yet, that only had the yellow and blue mutation. And all 5 probably originated from a source that had the blue mutation. We have time on the x-axis representing time between transmission events. So back to SARS-CoV-2, as you heard from Lawrence, the genome of the original Wuhan strain became publicly available very early on in the outbreak. Scientists across the world started sequencing strains arising in their local areas, and very quickly its natural mutation rate was calculated. So the rate was deduced to be at about 23.6 substitutions per year, and this fitted well within the range of mutation rates of other similar viruses, but it's much slower than flu and Ebola. This transmission rate for the coronavirus means that Cases might share transmission within a few days or weeks if there are less than 2 mutations between the two genomes, 2 cases. So, of course, scientists have kept sequencing. Thousands of genomes have been uploaded onto public databases, just as the gz.org that I'm showing you here. Also here I'm showing you a tree made from over 3,500 genomes that shows the emergence of strains over time across the different continents, and you can see the different branches. And in this case here, you can see the branch that emerged and spread all over Europe. In addition to trees, we can also look at the geographical spread of the virus. Here on the left, we can see all the transmissions that were occurring early on in the outbreak, stemming from China into Australasia, Europe, and North Africa and North America. But later on in the outbreak, we can see that the transmissions are spreading all over the world. So here in Coventry, we also set up our own sequencing service, and this was to parallel the testing facility that was being set up at the hospital in early April, and it was aimed to screen hospital samples from inpatients, outpatients, and hospital staff. This was a joint effort between Warwick Medical School and UHCW. So we got together the protocols already generated by our colleagues at Birmingham, and we bought the sequencer, which was the Oxford Nanopore machine, and with some data analysts from the university and a bunch of volunteering PhD students that, because of the later lockdown, they were unable to carry their own research in their own labs, came to help us. We set up the whole thing from scratch and started sequencing. And we joined a large group of other sequencing centers across the UK that were generating data that can help us understand the spread and nature of the virus across the UK. So now I'm going to talk to you about a small subset of our samples associated with a care home. COVID really had a great impact on care homes, with a number of deaths substantially larger than what is normally expected in an average year over the months of April and May. It was in the middle of March, the guidance from the government stipulated that patients with COVID-19 must not occupy an NHS bed if they're safe to be discharged. And it was not really until the middle of April, at the middle of this peak, that new guidance on residents from care homes stated that they would be tested for SARS-CoV-2 before being discharged from hospital setting. So really, overall, up to the middle of June, there were over 66,000 deaths of care home residents, and of those, over 19,000 were associated with COVID-19. It was right at the beginning of May when we were asked to focus our time in sequencing a set of samples that originated from an outbreak in a care home. At the time, we had no other information and proceeded to process the samples. Out of the 23 samples available for sequencing, we were able to only sequence 18. Sequencing is not as sensitive as the PCR testing or the other tests that you've been hearing from Nicole, and we need relatively viral loads to get good quality data out of our samples. The questions that were being asked at the time were whether the care home outbreak was associated with another outbreak at a local hospital, which was happening at the same time. And this was a hospital to which some of the care home residents had been admitted. We were also asked to see if there was transmission within the care home or whether all cases were independent introductions into the care home setting. And both the hospital and care home really wanted to see if there were any lessons to be learned for the future management of infections in the care home. as really the outbreak had cost the life of 1 in 3 residents in this specific care home. So we did a sequencing and we made our tree, and this is what it looked like. The length of the branches from left to right represents the number of mutations. And as you can see here in the red circles, we have groups of genetically identical samples suggesting spread between individuals. We only have 1 or 2 introductions into the care home. One spread widely throughout the care home, and the other one did not. Further analysis we carried out did not find a link with the hospital outbreak. I am not showing you this data here. At this point, we started gathering more information about the samples and looked at possible transmission events. So here is what a transmission tree looks like. Arrows represent transmission events, yellow squares represent a single mutation. I have also now colored the samples as blue for staff and red for residents. So it is clear now to see that there is transmission between residents and between residents and staff. It is not possible to distinguish who gave it to whom within the clouds of samples with identical genomes. And this is really one of the drawbacks of the relatively low slow mutation rate of the coronavirus. There could have been 2 introductions or 1, and we really do not have data to decipher this. We do not have a clear timeline for these samples, but all but 1 were collected at the same time during a mass screening that was carried out at the care home in early May. But we do know that a patient went into the hospital with COVID symptoms and tested positive in early April. Unfortunately, this patient passed away at the hospital and never returned to the care home. This probably explains why this variant was not widely transmitted into the care home. Another resident was admitted to the hospital a couple of days later, where he also tested positive. This resident was back at the care home in early May when the mass screening took place, and he still tested positive. Now, it is clear from this that the virus was circulating around the care home since March. Another interesting observation from this data set was that 60% of the samples we sequenced were from asymptomatic carriers, and this included both resident and staff. So, I mean, we're aware that this case study has some limitations. Not all residents and staff were screened. We didn't have samples from all of them. Not all positive samples were sequenced during due to the nature of the sequencing test. And samples were not collected, sequenced in real time. So really, the data represents a snapshot of the state of the coronavirus infection at the home in early May. And of course, the low genetic diversity of SARS-CoV-2 makes some of the data interpretation difficult. But we can still make some conclusions. Introduction was not linked to the hospital. It was probably community acquired. The care home banned visits in February. But of course, staff had to shuffle between their homes and the care home, making it likely that the virus was introduced that way. On the other hand, introductions, the restrictions did work, as there was a limited number of introductions into the care home. But transmission within the care home was quite large. The care home management stated that PPE was available at all times. But of course, taking into consideration the fact that our tests revealed that a large number of people were asymptomatic, the decisions the management had to make in terms of isolation and quarantining, it was really hard to do. So that probably led to the transmission within the care home. So we can make some recommendations. Actually, the current recommendations from England is that screening is offered to care homes every 28 days. I think some new recommendations are about to be announced, but I checked last night and they weren't available yet. But we think that our data suggests that a more timely approach should really be adopted. One of screening once a case is identified. In this case study, mass screening was not offered until 22 days after the first positive test. If screening was done early, then we should be able to control this outbreak much more rapidly. There's always a question of whether visits from relatives should be limited, or whether this should be screened before allowed to be visited, allowed to visit the care home. But we know from our data that in the case of the care home, that probably helped to minimize the number of introductions. But of course, considerations need to be made on how this affects the mental health of the care home residents. And of course, sequencing in real time, should be able to help us identify sources of infection and help with the control of the outbreak. And of course, acknowledgements. Lots of people have been involved in this project, setting up the sequencing service, both from the medical school and from UHCW. Thank you for listening.
Okay, thank you for that, Chrystala. We're going to move on. We've got quite a number of questions and comments, and we'd like to get through as many of these as we possibly can. So I think we'll just run through these in some sort of order, and I'll pass them out to people who I think might be able to answer them. I can start quickly. So Pete Briggs asks about the COVID-19 mutating and how effective any particular vaccine can be. Well, on the plus side, actually, this virus, as Chrystala has pointed out, doesn't actually change that rapidly. So the positive there is actually, it's not mutating. It's not like HIV. It doesn't combine or reassort its genomes like influenza. It has a relatively low mutation rate, but it is changing a bit. And I guess one of the interesting things, particularly about the vaccine that was announced yesterday, is that those nucleic acid-based vaccines allow you to plug and play. So as soon as there's any change that might be significant, that might impact a response to vaccine, you can change the payload, you can change the RNA very quickly. And in fact, you can immunize with more than one of those nucleic acids covering different changes. But everything we've learned at the moment suggests that those changes that are occurring are very slow. As for Nicole's question, Nicole Hamer's asked about the long-term health effects of RNA-based vaccines. Actually, we don't know. This is the very first time that an RNA-based vaccine has been developed all the way through to the possible mass immunizations. So we don't know, but everything tells us that it should be much safer than some of the other vaccines that are around. And I guess it's one of the risks that we're going to take in trying to prevent the disease. I think Wanya asked, and one for Nicole, about what is the simplest way to explain the science behind wearing a mask. Nicole might want to answer that one.
Well, I mean, to put it really simply, the most common mode of transmission of the virus is through little droplets out of your breath. So, and then the other way that you can get it is if those droplets land on surfaces, you can touch a surface, then you touch your mouth or your face, and you get the virus that way. So, wearing a mask can help prevent spread of aerosols, which is the name of these little droplets. And there's also an argument that it can make you more aware of what you're doing. So if you're wearing something, you're less likely to go around touching surfaces and then touching your face, for example. So I think that the evidence is quite clear that wearing masks can help. So that's why it's encouraged. So absolutely a good idea to do.
Thanks, Nicole. Cass Ford asks about COVID-19 seems to cause a wide variety and range of symptoms in different individuals, different sufferers. Is this rare, and do other viruses cause such a range of responses? Well, of course, other viruses do cause other tissue effects and other damage other organs, but there is something very worrying about this particular virus because it does seem to have an effect on our blood vessels, and it's that effect on our blood vessels which is very dangerous and seems to increase the risk of other diseases like cardiovascular disease and possibly stroke. And we don't understand why that is. Why does it make the blood more sticky? Is the virus actually directly itself infecting blood vessels? And these are things that are still being explored, but there is something very nasty about the virus, and that's why we need to get these vaccines working to block these other effects. Slangers21, can you explain how some people are asymptomatic while some people suffer with life-threatening symptoms? Who wants to answer that one? Nicole, do you want to have a go at that one?
I mean, I think the short answer is it's not really clear. So that it might be linked, and this ties in with another question that I saw there about the receptors. So we talked about the receptor on the host cell being the way that the virus binds to the cell, gets into your body. And somebody asked, well, why do you have receptors then, if the virus is just hijacking these to make you sick? So these receptors are all cellular proteins that have really important roles in how our bodies work. So, for example, the ACE2 receptor plays a role in a pathway that's to do with blood pressure and wound healing. So on its own, it's really important. And the virus is a bit of a parasite. It just binds and gets in via these important pathways. And one thing that's not clear at this stage, but there is a lot of research going into it, is that people might have different numbers and distributions of this receptor in their bodies. And for example, there was a suggestion that people that have diabetes or hypertension might have more numbers of the receptors that lets the virus in, although it's not. Just to emphasize, it's not a clear picture, but that's one example of why some people might react differently to others. Lawrence, if you want to add anything else there.
No, that's perfect. Peter K asks, when do you expect high volume 5-minute testing to be available?
Well, we're pushing as fast as we can. So we're working at the moment. We're trying to get it into the hands of those that need it as fast as possible. It's really dependent on how fast the regulatory approval goes, which is really important to make sure that everything that's brought out is safe and works well. So we're hoping that we'll have something out there in the next three months. But a lot of that is, like I say, is really dependent on getting the right approvals and getting something that works well.
Okay, there are a few vaccine questions which I'll answer. Michael Warner asks, do we have an idea of how long a vaccine will provide protection for at this stage? Weeks, months, or years, or is it still unknown? Well, of course, it's unknown. The bit of data that came out yesterday demonstrates that the Pfizer vaccine is very likely to prevent disease, but we're still not sure about whether that vaccine or indeed any of the vaccines currently under development will actually prevent you from getting infected in the first place. Lots of vaccines that we use, like the measles vaccine, for instance, prevents you from getting infected. We don't know that for these new vaccines that are coming along, and we don't know how long a protective immune response will last. But as long as it damps down the virus infection for a while, that should be enough in terms of generating enough immunity to start protecting the whole population. But this is complicated. It's going to depend on the nature of the vaccine, and it's going to be a big experiment, because when we start to vaccinate people, it's only then that we're going to be able to understand whether or not we can block transmission. But Catherine Widows asked about when the vaccine will be available. My view is that there's been a bit of hype about this over the last day or so. understandably so, because we have some light at the end of the lockdown tunnel at the moment. So people are getting overexcited. But I anticipate that it won't be till early in the new year that we really see the rollout of the vaccine. And as you'll have seen in the press today, that any vaccine, and there'll be others that will. We're going to hear some good news in the next couple of weeks about other vaccines, but it will take some time. And obviously, there's going to be a priority in terms of who gets it. It's true that Nicole says about the vaccine requires storage at -70. That's going to be a problem. The World Health Organization has done a lot of work with the Gates Foundation around cold vaccine storage and distribution in developing countries. The Pfizer vaccine does need to be stored at -70, but there are some approaches to try and improve that and increase its stability. It won't be the only vaccine, but it is a real issue for us in terms of the responsibility we have to developing countries. And some of the other vaccines may work better in that they won't require that so-called cold chain. And then Michael Warner's asked a whole lot of statements for us to consider, would you say? Michael's asked about airborne transmission is much more common than via surfaces, making masks more useful than wearing gloves. I don't know. Perhaps somebody else should answer that. Nicole, do you want to answer that one?
I'm not entirely sure if it's really, scientifically established which mode of transmission is more common. I think they're both really important modes of transmission, and they're sort of linked together as well, because airborne transmission deposits aerosols on surfaces, and then those can be picked up and transferred into other people. So I think the message is that where we've been advised to wear masks, is absolutely essential to do that, that wearing gloves isn't going to do any harm either. So I think both are important.
Michael asked about asymptomatics, and of course we still don't really understand what's going on there in terms of the fact that about 2/3 of individuals who are infected don't develop symptoms. Now, we don't know whether that's something to do with the load of virus, the amount of virus they were initially exposed to, the way people's bodies respond differently to infection, whether some people are more inherently protected than others. That's certainly true for children. We're beginning to understand why youngsters aren't as susceptible to infection, but we really don't know why some people are asymptomatic and how that's working.
An important thing there, though, is that asymptomatic people can still spread it.
Indeed.
Absolutely. So a kind of a mass testing approach, it's really aimed at picking up all those asymptomatic people, and then they can isolate, and you can stop the spread that way.
And that's a really good point, because one of the big problems up until now, it's only folks with symptoms who have been tested. And that's been a big problem for us, knowing that 2/3 of individuals who are infected are asymptomatic and still can spread it. So that's why this mass program in Liverpool is so important. And obviously we heard good news today that a similar mass testing program is going to be rolled out across Coventry. So that's great news. Michael also asked, do we know how many vaccines are ready to go in the UK from all sources and what the rollout plan timescales are? We don't know about the rollout time and timescales. We know that there are two other vaccines hot on the heels of the one we heard about yesterday from Pfizer. There's the vaccine from the Oxford and AstraZeneca group, and there is another RNA-based vaccine from Moderna. And I think in the next 2 to 3 weeks, we're going to hear clinical data from those vaccines. And then it's up to the government about how they're going to juggle the rollout of these different vaccines. It's going to be a real challenge, but at least we're going to be in a much better position. Okay. So any other questions in here that I've missed them? Should we be here's one at last for Chrystala. Should we be extra concerned with the mutation into mink in Denmark, and could we see leaps into other mammals? Chrystala, one for you.
Yeah, well, yeah, this has been in the news recently, the last few days. I think it's been a lot of a hype. The mutations that seem to appear in the mink and in the strains that were isolated from mink in Denmark. There is no evidence that they're making the virus more virulent or easier to spread or causing more severe disease. So the statements are not, they were a bit hyped in the newspapers. We've known that the spread in mink a while ago, actually, and they also spread in other mammals, if I remember correctly. Is that right, Lawrence? I think this has been identified in a few other mammals.
Well, actually, there's a couple of studies showing that the virus you can infect dogs, and some domestic dogs in China are infected. But whether they can spread virus, I think the worry with the mink is, is it spreading? I suppose. But
well, this is spread. It has spread to some humans, right? So it has been transmitted to some humans. But there's still some research to be done to really before we can say exactly what's going on.
There is, and it's exactly the type of work that Chrystala explained of doing that very detailed sequence that's so important. Is the vaccine at -70 degrees when it's injected? Elizabeth Turner's asked that question. Actually, Elizabeth, the way it works that people aren't talking about is the fact, of course, you can't inject a vaccine when it's -70. The vaccine has to be thawed, and it has to be kept in a refrigerator. And you can keep it all day, actually, in a refrigerator, and it's fine. So it has to be injected as a liquid. So I guess in all the concerns about the fact that you have to keep this really cold, when it comes to distributing this type of vaccine into GP practices, you can probably come up with some arrangement whereby you keep it refrigerated for the day before it's used. I think that's all the questions, and I think we've just run over our time, but thank you everybody for sticking with us. I think it's been a great session. We're very very grateful that you've stayed with us, that you've asked some really interesting questions, and I hope you found this whole session informative. We're going to be holding more of these Science on the Hill online events across a range of our different research programs at Warwick. So please check back on our events web pages, and you'll be able to see what's coming up next. So again, thank you for watching wherever you are. Please take care, stay safe, and remember that whilst vaccines are coming along, we still need to keep our distance, wear masks, and wash hands. So stay well, keep safe, and goodbye for now.